So we will have we do not need that OCR actually.
Let me introduce you. This is our lab coordinator.
Let them have a seat first.
Okay.
Get comfy. We got enough seats for everyone. I hope so.
Okay. Let me introduce you. This is our coordinator lab for physiology. Dr. Nazwinya Bar. He may prepare you with some samples and also equipments and facility that we have in this lab.
Yeah.
Nice to meet you.
Nice to meet you too. One second, I need to get this thing out of the way. Right. So, welcome to physiology, or should I say, half a day. It's not every day we get a visitor from, unusual place on the planet. Right. Okay. Where did you come from? Did you go?
We are from four universities. She is assistant professor.
Okay. All right.
University of Guam, and she has 3 students. Green color 3 students.
So, are you all postgraduate students?
Professor undergraduate.
Undergraduate.
And we have another group from Tennessee State University.
Why this isn't?
Student, and another 2 are faculty.
All right. Okay.
Professor.
All right. Okay.
And I'm from Northern Marianas College. These are my students, and I'm faculty there.
Right.
And we have one student. She's food science.
Food science. Okay.
Food science from Washington State University.
Right.
This is our collaboration project.
Very high diversity.
Yes.
That's good.
And it's like extension.
So, how do you like Malaysia so far?
It's very great.
Or is it too warm? You're tropical, somewhat region, right? So this should be. But I guess I think this month we get less rain compared to other months. Yeah, if you come towards the end of the year, that's when the monsoon hits. Then you'll be right. Oh, I need different umbrella for different days. Right. Right. Okay. So thank you for coming. So we'd like to see what we have here in our physiology lab. So I need to get this done. So if some of you are undergraduate, right? So if you're not familiar with physiology, I think you are very familiar. It's basically we learn how the plant function, okay? Because plants are just like humans. We are living organism, so we have functions. We have organs. Plants have organs too, but not in the similar manner that we have. Okay. For example, we human, we have glands. Okay, we have pituitary glands. But plants do not have such thing. But somewhat there are some overlapping of functions to produce similar result. Okay, so we understand how the plant functions. So in the context of agriculture, this will be very helpful to improve crop, especially crop production, right? Because when we understand what functions of the plant that needs to be revved up, we can focus on that and feed the information to people that do the breeding, right? So breeding is very important. That's where we could get our good seeds, very vigorous plants, right? And pretty much when you do the breeding, it will come back to physiology, because the breeders need to know what to be tweaked. Usually when it comes to plant production, crop improvement, it will go back to photosynthesis, because that is the fundamental food manufacturing process that will have impact on the final yield that we harvest, right? Okay, and also with physiology, we also able to understand the biotic and abiotic stresses on the plants, because just like us, plants are very dynamic. They interact with the environmental factors, the wind, the temperature, even the earthquake. Some places. The good thing is Malaysia is slightly outside the Pacific Ring fire, so we don't really get the earthquake. We got the tremor from time to time, but nothing serious so far. It's a blessed land, somewhat. Right. Okay. So and finally, there is a post-harvest bit. So this what happens to the produce after you have harvested them, because many crop you can't immediately sell to the consumers. Okay, so you need to have some kind of system to enable the plant or the produce to be paused so that it can reach the supermarket, and then you as consumer can buy and enjoy them. And that also requires the understanding of plant physiology, what can be done to prolong the shelf life, right? If you put everything, you might think, okay, this is all sounds like a business, money making. Yes, it is a business. Yeah. Although if you go back 1,000 of years ago, 10,000 years ago, during the caveman, no, 10,000 years still, caveman way before that, they don't really talk about money. They don't talk about sustenance. So, but that's why I like to call agriculture is the mother of all sciences, if you think about it. Because with the activity of agriculture, the people of the past, your cave ancestors, they need to learn about how the plants function, the soil, the nutrition, the water, even the climate. They need to understand that. So due to agriculture practice, all these other branches of sciences come to existence. Interesting, right? Right. Okay, so today, just very quickly, I would like to highlight three our highly sought after instruments in our lab. So we have the chlorophyll meter, spectrometer, and portable photosynthesis system. Have you seen this before? No? These two actually from Nebraska. You know Nebraska? Have you been there? Yeah. It's a corn state. Right. So the first instrument is this guy. So this is what we use. Oh, I'm sorry. This thing is playing music. I should shut that. While waiting for you, I need to soothe myself, like soothing. As it get warmer, you get angrier. Right. Okay. So this is the SPAD meter or the chlorophyll meter. It is very good to measure not directly the content of chlorophyll. Chlorophyll is the pigment that gives the plant its green color. Therefore, it's very important in photosynthesis because chlorophyll is the very protein complex that capture the photonic energy from the sun and then turn it into the sugar, and then eventually the fruit or leafy vegetable that you consume on daily basis. So this thing, it measures the relative content of chlorophyll and by extension, the content of the nitrogen as well. So it's very easy. It's non-destructive, meaning that your plants do not have to die in the process. So if I turn that, it's very easy. I'll just do the clipping here. And then what? Calibration error. You have that? Yeah. Right. I'll just use this plant. This is cat's tail. Right. So it reads here 48.7, but it is without unit. It just tells you the relative absorbance and transmittance of the laser. So you can see here, this instrument, at the top here, there is a laser emitter, two laser of two light wavelength, 650 nanometer, which is the red light, and 920 or 940 nanometer. 650 nanometer is the red light absorbed by the chlorophyll. So if the leaf that you clamp on has lots of chlorophyll, the absorbance will be high, so that whatever transmitted to the other side, to the bottom, which has the detector, will be much less. The other wavelength, 940, is to measure the other structure in the leaf. It can be water, it can be veins, any other structure other than chlorophyll, right? So knowing these two values, there is a microprocessor in this detector that will calculate and give you the reading. So the good thing is, it will tell you, without killing the plant, is the chlorophyll abundant? Because if the chlorophyll abundant, meaning that you have given the plant enough nitrogen. Therefore, do not fertilize further, because nitrogen is input. Okay, so again, business, you need to play around with your input and output, right? And if you want to go slightly further, you can do this regression where you send your clamped leaf to the lab so that you can quantify the chlorophyll content. And this can be quantified using the equipment such as spectrophotometer. We have the one at the back here, but that's very old. I hope that gets changed soon. And so that next time when it reads here 40, for example, you will know what is the value in terms of milligram of chlorophyll per square meter, right? So the magic of regression, right? Okay, and the second is our spectrometer. So the story behind it can be rather complex because we're talking about light. We as people, agronomists, horticulturists, sometimes we don't see light as input for our agricultural produce. But actually, light is something need to be understood if you want your agriculture produce to perform very well. So in essence, if you learn your physics during your, perhaps you did your A level, light has dual properties. Okay, it has the wave properties and also the particles properties. Okay, when we're talking about light in the sense of particle perspective, we are talking about the light intensity, how bright, how dim it is. Okay, because the more particles of light present for a given area, the more intense the light is. And the second property is the light quality, which refer to the wavelength, and that we can see by our eyes because that is the color. Okay, so the wavelength of the light correspond to specific color, and this color of specific wavelength can be detected by our retina. You know that we have a tissue back of our eyes, we got photoreceptors, and our photoreceptors can perceive these signals and turn it into colors that we see every day. For example, if the light coming to your way, the wavelength is around 400, you will perceive it as blue color, right? But if the wavelength slightly longer and therefore slightly less in energy, it will come to your eyes and your retina photoreceptor will perceive it as red color. Yeah. Interestingly, our retina, our eyes photoreceptors only sensitive to a very narrow band of light wavelength between the 400 nanometer to 700 nanometer, and that coincides with the color of the rainbow. Yeah, our eyes like rainbow. Other than that, we can't see. Yeah, although there are some people a bit Newton in their eyes. You can know if you ask your friend, what's the color of the sky? If he or she answers violet, that's the Newton. Yeah, some people have that mutation in their eyes receptors. Yeah, not disabled, just gifted. Yeah, yeah. He might be able to see your long lost ancestors floating around the house. All right. Okay. Oh, sorry. I want to demonstrate this. So if if I turn it on, I'll just hold it. I have the screen here. How do I? Yes. Let me bring it up there. Yeah. So now we would like to know the light in here. And what do we do? I'll just connect this to the computer and then I can capture it so we can know what's hidden in the light here. It looks dim, right? Is it capturing? Okay. Yeah. Yeah, so it should be this one. Yeah, not this one. I need to, yeah, so this is the current light quality of this room. You might think this is far too dim, right? But your eyes can be deceiving. Heard that before? Because in terms of proportion of colors, of color ratio, in our environment now, in this lab, there is a dominance of blue color. And that's known to happen for the white light, the fluorescence light. Okay? And very low amount of red light. Okay? And we got some green light. Okay. Right. But what if I use this UV color? Is it going to be the same? Put it under there. Ready? Okay. So let it read. Can you see that? There is no other colors present, but there is a peak of reading in the UV region. UV region is whatever before 400 nanometers, right? Okay, so it's a pretty cool instrument. You can play around with it. Even if I want to read my screen got blue light protection. Is it so? That's what the manufacturer claims. Well, not so much because we still got blue. Yeah. So why this is important? So, like I said, light is an input for agriculture so that you can understand when your plant is doing well and you would like to imitate the lighting situation somewhere else, you take this instrument, spectrometer, get the light reading, jot it down, and then when you go to the new place, do something to achieve the same light quantity and quality. And then that's how you can be homogeneous in terms of your crop production, right? Okay, and finally, it's this, let me get that out of the way, is this guy here. Ta-da! And that's the most expensive thing that we have in our lab. It's costing, this is LI-6800 portable photosynthesis machine. We got one here, you can have a look later. How much does it cost? If I use USD, it's about 120 grand. Or with the tax, with the tariff and everything, thanks to Mr. Trump, maybe 150 now. Yeah, so its job is to measure photosynthesis. That's the basic of everything, without which we wouldn't be having this discussion. Right, remember photosynthesis? So that's your, what's this? What's this organelle? Chloroplast, okay? So for photosynthesis, it has two reactions. We have the light reaction, from which you'll get ATP, NADPH, and also the byproduct oxygen that we breathe in. And the second reaction of photosynthesis, popularly known as the Calvin cycle. From here, you get the precursor to sugar, or popularly known as glycerol 3-glyceraldehyde. Yeah, or there are many names. Okay, so what this instrument is able to, it can measure each of these. It can measure the part of the light reactions, and also it can measure the part of the Kelvin cycle, right? Because you can choose the mode, right? If I can show you the screen of this instrument, is it there? Yep. Drag it. I like this function to be able to drag the screen. One thing I don't like to drag is my feet to work. Not just me, right? Everybody don't like to do that. No, I don't drag my feet to work. I enjoy being a plant scientist. Your world is very colorful. You see the lights are so colorful. What else can I see? Right? Okay, so now this instrument is connected or is clamping this particular plant. A califa is the cat's tail plant. You can see the catkin here, the inflorescence here. I can plug this right there. That souvenir from UPM. Like. So, what does it tell here? In this screen, you can see that there are many pages, 1 until 8, right? For this page, it tells you currently the photosynthesis reading for this plant is around 7 micromole of CO2 exchange per square meter per second. Meaning to say, if you remember your Kelvin cycle has 3 phases. You got the carbon fixation, you got the reduction, and also the regeneration. Therefore, this instrument measuring the first phase of Kelvin cycle, namely the carbon fixation, right? And then what else do you get from here? You also get the GSW. This is the stomatal conductance. How much the stomata transpire the water loss. Meaning to say, if this value is very high, meaning that the plant is not water use efficient, and that's probably something that you try to minimize. You need some amount of stomatal conductance, otherwise your plants, the leaves, are not able to do the gas exchange. You see the leaf, like the leaf here, can I plug you? I'm sorry, for sign, don't scream. Your leaf here, underneath your leaf side, there are many microscopic pores called stomata. And stomata are like mouth. Literally Greek word, it means mouth. Open and close, so that gas exchange can happen. Why? When you do the photosynthesis, there is a byproduct, remember, the oxygen? That oxygen needs to be used so much by the plant, and there is an excess. Too much oxygen inside the leaf will be detrimental to the plant. So the plant needs to allow the oxygen to escape, to exit, while in the same time allowing CO2, carbon dioxide, to get in, because CO2 is the ingredient to do photosynthesis. But the truth to the reality is, not only oxygen exit and CO2 enters. In the process, water also escapes, and water is a precious commodity for the plant. You see, the plant, it doesn't have a skeleton like you. It depends on the water potential, water pressure, hydrostatic pressure to stay firm. When it loses water, what will happen? It will start to wilt, right? But it has to open the stomata to allow the CO2 and oxygen exchange. So there is a continuous dilemma of the plant. So that's why understanding the stomata control and movement is one of the interesting field in agriculture, even in general biology. Because if you know how to control the stomata, you can control water cycle and carbon cycle throughout the planet. Right. Okay. So this machine can tells you that. What about other pages? Okay. Depending on the modes that you choose, let me show you. You can also know the temperature of the leaf. Right now, the leaf is around T leaf, meaning temperature of the leaf. It's around 27.6. And also the temperature of the instrument itself. What else? Yeah, I forgot to mention about this actually. The RH. Yeah. So basically, you can measure anything that is pertaining to the plant. All these environmental inputs. The flow, which is the wind. The air, the RH, relative humidity. If humidity I think this is important for you as well because you are somewhat tropical, right? If the humidity goes below 55% relative humidity, the stomata are going to close. You're not going to get photosynthesis, but you're going to get photorespiration. So I can see that many people in the rather hot region, they are struggling with this because they don't understand this concept, especially in the glass house. Yeah, you need to control the RH to be in the sweet spot between 60 to 80. You don't want too much because if it's too much, it's going to condense and fungals will start to come, right? And then what else? You can control the CO2 in here, meaning that this machine can somewhat predict the future. You can't get, Back to the Future movie? Yeah, you can't build the machine to know how this cat's tail plant would respond in 2070. But based on the scientist projection, you know that in 2070, the CO2 level is going to be 800 ppm, for example. No need time machine. You just tell the machine here, change the CO2 to 400 or 800 micromole or ppm so that it can, I can change it here. Oh no. Keep forgetting. Yeah. So you can change it as you please. Yeah. And then you can know what will happen to the photosynthetic rate, what will happen to the stomatal conductance rate without having the time machine. So it's very useful actually to do the prediction with this machine. It kind of simulate, right? Okay. And what else? You can play around with the fan. Why do you think there is a wind in our nature? You go to the beach, Hawaii, your neighbor, remember? People go by the beach looking at the coconut tree, blustering of the wind. It's moving very, very elegantly. Why plants have to move along with the wind? Why?
Can you finish it in 3 minutes?
Yeah, sure.
Next station.
Almost done. Yeah. Yeah. Because to break the boundary layer, there's a boundary layer here, and without breaking it, there is no CO2 going to get in and there is no photosynthesis. So wind is very important so that you can enable the plant to break the boundary layer and enable the photosynthesis. And also you can control the temperature. And this instrument, you can plus minus 10 degree from the current. If the current is 30, it can go as high as 40 degree and as low as 20 degree. And of course, the light. Like what I mentioned earlier, the light quantity and the light quantity and quality, you can change that as well. Right, so yeah, that's pretty much that. Okay. Somebody called me prof. That sounds wrong. All right, okay, that's all. If you want to read further, I have a collection of video. You can have capture the QR. You can watch later. All right. Just a quick disclaimer: don't go to the weird playlist in this channel. Go to the educational playlist. All right. Okay. All right. All right. Okay. Thank you. Thank you very much. Okay. All right. Good. Oh yes. Any question? Yeah.
Is the mitochondria the powerhouse of the cell?
Yes, mitochondria is the powerhouse of the cell, and they work in tandem with chloroplast. What the chloroplast has produced, the mitochondria will use, break it down, and you get their energy. Yes. Yeah. Usually, you have a healthy chloroplast, you will have a healthy mitochondria. They're interrelated. All right. All good. Turn on the light. Thank you. All right. So where are you going after this? No idea. All right. Okay. All right. So I'll see you around. Have a good trip. Enjoy your time. All right. No problem. All right. Thank you. That's your souvenir. Or immigration might. Yeah, sama. Yeah, you're welcome.